Yeah. Thank you, Ms. Leena. Thank you so much. At the outset, I would like to welcome our great eminent scientist and guest of speaker for today, Dr. Pradeep Mahajan, Chairman and Managing Director of STEMRx Bioscience Solutions Private Limited. A warm welcome to you, sir. And 1 minute quick note about this wonderful weekend webinar series. We started this with 3rd October. So this is a session 2, and we are so happy that we've got an overwhelming response from all different parts of India. And this theme is stem cell biology bench to bedside. So under this theme, we just started with the introduction to stem cell magic of stem cells, which was given by Dr. Ramesh Bonde, Research Director at DPU. And today's session, session 2, here we have a great eminent scientist and the guest of today's session, Chairman and Managing Director, Dr. Pradeep Mahajan, to enlighten you all for this wonderful session. And he's going to talk on mesenchymal stem cells, living drug molecule. And he's going to elaborate and enlightening you with this wonderful molecule, mesenchymal stem cell, and all its applications. And once again, thank you so much to FABA for having the affiliation with IABM. It's a great pleasure for us. And my sincere thanks to all participants for doing a great job in being a great part of it. And with this small introductory note of IABM webinar series in affiliation to FABA, I request Ms. Leena to proceed.
Thank you, ma'am. Now I request to Dr. Pankaj Kaingade to introduce our today's speaker of this session.
Thank you, Leena. Am I audible?
Yes, sir.
Yes, you are.
Yeah, thank you, Leena, and thank you, Dr. Indu, for getting me in this webinar series. Also, now we are in the second session, and I'm really happy to introduce Dr. Pradeep Mahajan. So let me just start my screen. So here it goes. So, Doctor, thank you, sir, for being here. So I'll introduce Dr. Mahajan. So Dr. Pradeep Mahajan is the Chairman and Managing Director at STEMRx Bioscience Solutions Private Limited, Mumbai. He has a very brief CV. I'll just go through it shortly in this CV, so we can quickly start the session. So Dr. Mahajan has completed his Masters in General Surgery from Marathwada University, Maharashtra, and went up to pursued Diploma in Urology at the University of Vienna, Austria. In a career-changing move, after 3 decades of being a successful general and neurosurgeon, he started his own brainchild, STEMRx Biosolutions Private Limited, in 2011. This was to focus on in-depth research in the field of regenerative medicine and cell-based therapy, which he believes is the solution to address the limitations of conventional therapeutic models. Dr. Mahajan is also a life member of the and Joint Secretary of the Stem Cell Society of India, as well as a Vice President of Anti-Aging Foundation, Delhi. Also, he is a Scientific National Advisor of an Indian Stem Cell Study Group. He has also been appointed as an Honorable Associate Professor of Regenerative Medicine and Cell-based Therapy at Dr. R.N. Cooper Hospital, Mumbai. Also, he is on faculty for MD Transfusion Medicine at KIM Hospital, Mumbai. Also, he has been an examiner for various fellowship courses at our Diva Patil Institute, Kolhapur. He has total 13 publications to his credit, also various patents, international patents for treating of avascular necrosis using regenerative medicine approach, and many more. So now I hand over this dais to Dr. Pradeep Mahajan, sir. Welcome you, sir. You can go through your presentation. I'll stop my sharing. You can share your screen now.
Thank you so
Over
much
to you, sir.
for the introduction. Now may I request our guest of honor, eminent scientist, chairman and manager director of STEMRx Bioscience Solution, to start the session. Welcome, sir.
At the onset,
I'm sorry. Host, can I change the host rights to Dr. Pradeep Mahajan? We can't share our screen.
Ms. Leena, please make Dr. Pradeep Mahajan screen Dr. Pradeep Mahajan as a host.
Co-host.
Yeah, please make Dr. Pradeep Mahajan as a co-host. Thank you.
He is already.
Yeah. Thank you.
Yeah, the screen is visible.
Dr. Indumathi, let me congratulate you before I start my talk
Thank
behind
you, sir.
of my this series for taking an initiative in academic program. That shows the interest of you pursuing the academics of neuroscience. I'm much impressed for one reason. You are taking directly the research people from bench to the bedside students which are coming up, particularly in the biotech technology. Now, if you, and also I see that there's a lot of work on site yours. You have collaboration with the Kolhapur Cancer Center and Federation of Asian Biotech Association, which are
Asian Biotech Association.
wider scope. The theme of talk, which is stem cells, more important, bench to bed. Now, what does it mean? Bench to bed means the science today, what we are learning in medical school, it means that has been taught 50 years before researched out, not today. So you people are so lucky, you are getting an exposure of the science which is right now in the benches, which is coming to the beds. Now, otherwise, as an academic in a medical school where I teach as a professor of surgery, I have seen the academic teaching today, which is researched about 50 years behind or last 50 years before. Now and beyond. The hope and beyond means when I talk of the word hope, it indicates that there is a hope for everything which new comes. And beyond, yes, beyond the drug molecule, what we are using today is a non-living drug molecule. That is what I call a living drug molecule and mesenchymal stem cell. I have a concept of belief. How do we change the science of pills to science of cells? You all being students, there's a big curiosity. Can we change the science of pills, what we prescribe on today, to the pills? Yes, that is what is very important. This being an academic meet, my entire dedication will be an academic talk, which is very important for you. What is being taught today in medical schools, even as a professor, what I teach in the medical school is not what I practice today. That is a big difference, or that is a big understanding what I would like to bring to you as a students. Infertility, concept of infertility, concept of cancers, concept of diabetes, concept of autoimmune diseases, concept of autonomic nervous system, everything is changed, and which I have been teaching for last 30 years, the same thing what was researched before 50 years. So you guys are so lucky that this academic program, you should thank Dr. Indumathi bringing directly the is happening on a flat cell is the next pillar of medicine. Let me share you the next pillar of medicine. This today's talk is going to enlighten you in a very different aspect of cell as a molecule when we call as a drug. What is the difference between a molecule, what we prescribe today, and what is the difference between a drug molecule, which is living being, that is living drug molecule. I call it to be a master cell. In most of my talks, I always believe in a concept of you carry your repairing kits in your own body. You carry your repairing kits in your own body. Does it indicate that there are repairing kits in your body? And these repairing kits are your master cells. These are the master cells. By training, if you see, I'm an absolutely allopathic surgeon. Till 9 years before, if you would have asked me, I would have not believed science is running so fast changes. The moment I changed from my surgical career to regenerative medicine or cell-based technologies as a researcher, my all concepts of medical practice have changed now. The thought process as a surgeon and thought process as a researcher are two different concepts. And a newer teaching, the clinical team has to shake hand with the basic researchers so as the biology will become molecule. How many of you know MSC as a mesenchymal stem cell? It is a stem cell. Can any one of you audience students can tell me whether MSC is a stem cell? This you can send me an answer later on. Reason being that when I started reading regenerative medicine maybe 9 years before or 10 years before, then MSC was not understood what MSC is understood today. Means it is dynamic. What I'm going to talk to you today may not be true for tomorrow, may be changing tomorrow. Reason is MSC is just before 2 years, we used to talk as a stem cell, but today in my talk, I will prove you MSCs are not stem cells. They are medical signaling cells. During the course of each slide, I will have a presentation to take you to understand mesenchymal cells are the medical signaling cells and not the stem cells when we talk in vivo. In vivo means within the body. And when we take them outside the body, that is in vitro, yes, they can be stem cells. See the two contrast end of a magic cell. Within the body, it's a medical signaling cell, whereas the moment you take this cell out of the body, it becomes a stem cell. See, this is a big difference which probably in the last 2 years has changed the concept of MSC. Dr. Arnold Kaplan, we call him as a father of mesenchymal stem cell. He was the one who defined in 1990s bone marrow do exist with one more type of cell than hematopoietic stem cell. Even today, in a standard medical textbook in a medical school, when we teach bone marrow, we don't have any reference in the text of MSC in the bone marrow except the recent one publications of 2019-2020. Otherwise, if you see the publications before 2019, all of them will show you in bone marrow, there's only one stem cell, that is hematopoietic stem cell. That is what the dynamics of the medicine are there. That is what the dynamics of regenerative medicine are there, the stem cell are there. Every year, that's what I told you during my initial talk itself. What I'm going to talk to you may not be true tomorrow. If the same talk I would have given a year before, I would have been different way presenting mesenchymal stem cell. Today, I'm going to present in a different way. Maybe tomorrow it will come with new research things about the mesenchymal stem cell. The limitations of the healthcare management today are for reason. I always tell in medicine, what is being practiced has got a lot of limitations. If you see overall, there are a number of conditions. When I used to practice as a surgeon, I used to tell my patients, this is not possible, we cannot do. Like autoimmune diseases, we cannot do anything. Diabetes, we cannot do anything. Once you go in insulin, we keep on changing insulin till it comes with a kidney failure or retinopathy. Cancers, we used to teach something, now the concept of cancer stem cell, which is very different than the cancer cell. Infertility, concept was different. Now I treat human body itself is a bioreactor. We don't require laboratories. Your body itself is a bioreactor. When we have a mesenchymal stem cell, the body itself plays a major role of different environment, different functions of the MSCs. Whenever new science comes in, we require its acceptance in the community, its acceptance in the practicing specialists or facility or professionals, which is very important, which we'll see today how its presence has been identified. And the global scenario, how it is going to be a changing next leg of medicine after maybe another 10 years. I have a simple one hypothesis when I teach most of them. This hypothesis is individual cell of your body is an individual functional unit of the life. Individual cell of your body is the individual function of the life. And all of them come together to make a one multifunctional life. Multifunctional, that is human body. Now, when I say each individual cell is a small structural functional unit, means it has got its own individual function. And if individual small functioning cells come together, they make an office, that is an organ. That is where the homeostasis is very important. Then cell has to communicate to each other. And if this communication is homeostatic, the organs functions well. This is the basic concept of body function. Any deregulation in any kind of cell function, quantitatively, qualitatively, will lead to a dysfunction of that organ, and that dysfunction is called as the disease of that body organ. Now, when we talk individual cell to cell, it means they need to be individually perfect functional. Now, these MSCs, which are very importantly called to be an individual cell, they do have a functional limitation. And that limitation we'll see subsequently why we want to overcome this limitation, use exogenous source of MSCs to convert this body repairing system into therapies. Now, the conventional medical system can only do a corrective job of signs and symptoms. It cannot repair the body. If you see what I have given here, it has got a limitation. It can only elevate your signs and symptoms, nothing beyond that. The further, whatever the repairing takes place is done by the internal repairing system of individual cells and not by the drug molecule which I prescribe you during your illnesses. They have their own limitations, and overtaking all these kind of medications in the subjects which I told you, like autoimmune diseases, diabetes, cancers, these drugs have their own more side effects than the benefits to the patient. The classical two examples which I have given you here are very, you could understand important of them are insulin and nitroglycerin. If you see insulin and nitroglycerin, we have been using them for years together. Insulin, if you remember, 50 years. Nitroglycerin, if you remember, 100 years. Ever have you seen the patients who are using insulin for the diabetes or those who are cardiac problem using nitroglycerin? They have overcome a repairing system of the body and your organs were repaired. They only treat the signs and symptoms. They cannot repair them. This is what today's talk is going to make you understand. These drugs, what we use, they only manage to treat your signs and symptoms. And what we require are the master cells, which I just was showing you about the stem cells. But MSC in vivo are not stem cells. They are signaling, medical signaling cells. So we need to change the paradigm now. Our own health system we have to change. Thinking from a pharmacological molecule to a biological molecule. A big paradigm shift in the healthcare from a pharmacological molecule to a biological molecule. Heading not to treat the signs and symptoms, but to repair the systems with minimum side effects and complete safety assurance. And these benefits, what we are going to look through these therapies, which I'm going to talk through MSCs, are the long-term benefits, not the short-term. So in overall, if you place right thing at right place at right time, the effects of them, what we'll see are the regenerative. This slide, Albert Einstein always try to talk on this slide with everyone for one reason. We can't solve the problem by using the same kind of thinking we used when we created them. It does mean, remember, it does mean if I want to bring solution to the topics like autoimmune diseases, cancers, diabetes, or any other disease of that kind, let me share you. If I am to work on pharmacological molecule which last 100 years, when the antibiotic era started, we just celebrated 100 years of antibiotics. Penicillin when was introduced in the market. If you are going to use that pharmacological molecule, we will never will find a solution. So we need to change our basic concept of healing, repairing, and not treating the signs and symptoms using pharmacological. That is what he says. He says if you really are looking solution for unsolved questions, then if you don't think in a different way, you will not have solution to this problem. So we have to look into disruptive innovations. Can someone assist me to roll slides? Sorry for interruption. What's your time? Sorry. So we need to have innovative concepts which are disruptive. The word disruptive is here very important because when I'm going to prove you today so many concepts of medicine, these concepts of medicine are still in innovation. They are still in the research. They are reaching to the benches, from benches to beds, and we have to prove the disruption of the innovations which were existing and bring in the new ideology of treatments, breaking the conventional thinking. So this distribution will bring you closer to the nature for the better of the mankind and proving beyond the proven things. And that is done by regenerative medicine. And this concept is an overall umbrella which has got a role to play to regenerate, restore, repair, and replace. Now, all these four functions which I have shown here, though I have shown lower down all other hepatocytes, autologous cells, beta cells, VEGF, PRPs, in spite of that also, MSC alone in vivo plays a major role of regeneration, restore, replace, and repair. That's the beauty of MSC. And all these things we can achieve for one purpose, as I was talking to you, repairing the organs, restoring the functions lost, and bringing the organs as they were before they were damaged by any kind of inflammation, bacterial, viral, traumatic, or mutations. We have to see that they repair back to the treatment before the illness, get them back to us. And all these things require surely a lot of involvement of science, of basic scientists. Politicians have to support, wealth is required for the research molecules. And these research molecules, without having backbone or from these kind of strong social backgrounds, we cannot bring it to the benches. Now, luckily, if you see, we are in a phase where last 10 years, where most of things were challenging, even in a main line of medical stream where I used to practice, I had to face a lot of challenges to make my own colleagues to understand. Now we have overcome, because if you see globally, likewise the way we have today a webinar, there are many institutional programs, individual associations like surgical associations, gynec associations, or orthopedic associations. In every association conferences, you will find one dedicated talk on stem cells or regenerative medicine. That itself speaks that there's a good amount of acceptance. Now coming to the topic of mesenchymal stem cell, when you define as a stem cell, as I told you 2 years before, we used to call it a stem cell. The specificity, it has got 3 characters expressing CD are the cluster of densities which are present on the cell surface, that is 107, 90, and 133. which should not express 34 and 45, that is hematopoietic, and HLA-DR should be absent. And one more basic concept is it should be adherent to the plastic vessels. Here I have forgotten to write one more. They should in vitro have a basic differentiation in three particular molecules, tissues. One is adipose, second is bone, third is cartilage. In clinical practice, when we talk about MSC, I call it to be a king of all the cells. Now, when we use cells coming from the community of immune system, when we use cells coming from the basic stem cell community, and cells which are adult cells or induced pluripotent cells, in all these type of cells, globally, if you see, MSC is top in research as a research molecule. If you see the clinical trials globally, there are 7,000 plus clinical trials on MSC as on date. If you go and Google on clinical trial GOV, you will find more than 7,000 clinical trials on MSC alone. So I call it a very innovative drug molecule which has a very multifaceted personality. It has unmatched qualities within the body, that is in vivo, and outside the body, where in vitro, it plays a major role as a molecular source of various bioactive molecules, growth factors, and recently during my course of talk, I'm going to add one more concept of MSC, which is a very powerful as an exosome, source of exosomes. And the best part of these cells compared to any other cell is when we want to talk it as a drug molecule and you want to bring it in the commercial market for therapies using this cell as a drug molecule, then we need to see, can we scale up their manufacturing process as a robust methods? So far, they are the best one where we can talk of converting them, making the cells into from billions of cell manufacturing by using robust technologies. How many of us know that our body has got a very strong regenerative potential or innate regenerative capacity? Are we aware that our body produces red blood cells, 50 million cells per second, every second? How many of us know? How many of us know our skin every 2 weeks is replaced? Bones replaced every 20 years. It indicates what I was talking to you when I started at the starting of my talk, that we carry our repairing kits. Means every adult cell is replaced by a new stem cell, that is resident stem cell of each organ. The organs have their own new resident stem cells and not the MSCs. MSCs play a different role and resident stem cells play a different role. This is what has happened today when after 1980s, the adult bone as the source of stem cell only in the bone marrow. Now 1990 onwards, the Arnold Kaplan, whose name I just referred sometime before, he defined existence of one more additional all type of, I call this to be a messengeric process. When you take this cell, MSC, from in vivo in a petri dish of your laboratory, I promise you can make this MSC dance the way you want. You can differentiate it into any potential differentiation of a tissue, and mammalian cell, adipogenic cell, and you talk of a cell there's a lineage which we can create from a MSc in a lab, from top to bottom, if you see, at every stage. And this is a role of tissue engineering. MSCs play a major role in tissue engineering and tissue developments using MSc and various signaling molecules in between, like vitamin C, steroids, Ds, and various CD molecules, and give them into different specific lineage and different tissues. And this entire messaging process makes a wonderful presentation of today's talk, how beautifully MSCs can change the future of medicine at every aspect, from a simple molecule of treatment as an biochemical or biological product to a cell component. Always talk of hypothesis, like earlier I spoke about the hypothesis of cell-to-cell communication. Now I'll tell you a second hypothesis, which is a Chang's hypothesis. In this Chang's hypothesis, what he says, these cells are pericytes, anchoring around the capillaries. Now, if you remember, I told you sometime before during my course of initial talk that autonomic nervous system, which we were taught, and now the blood pressure which is managed by autonomic nervous system, we used to think that autonomic nervous system is there, which plays a major role of blood vessel contraction, relaxation. Now it doesn't no more exist. Exists that these are the pericytes which contract the capillaries and relax. When the blood pressure shoots, the pericytes contract the capillaries and manages the blood pressure. Now these pericytes, as I told you, could be a very beautiful presentation in every therapeutic index of body repair system. When any injury takes place, these pericytes de-anchor from the capillaries under the influence of PDGF, and they get into a immature to mature MSCs. And then these MSCs, they play a role, not a role of stem cell, but they play a role of medical store, which I have shown you earlier. As I told you, they will play a role of secreting various molecules depending on the environment in which this reaction has taken place, where the injury has taken place. Now, this hypothesis says MSCs, the moment they come out of the anchoring of the capillaries, they have various tropic effects, immunomodulatory effects, antimicrobial effects. Now, they come through various signaling molecules which are secreted by these MSCs. Now, MSCs, here I'm talking as a cytokines and chemokines. Subsequently, I'll show you a different role of membrane molecules, which are exosomes. How do they exist? And there comes the growth factors again. All a range of 800 plus secretory molecules which come from these mesenchymal stem cells or mesenchymal cells. And the moment they finish their job, if you see the hypothesis shows here in the slide, you can see the job is done of a healing through the tropic effect, immunomodulatory effect, antimicrobial effect in injury. And the same MSC goes back in a re-established endothelial cells, making new capillary tubules and anchor around the capillaries to stabilize the vascular system of that or damaged organ again. See how beautiful microenvironment is changing from a repairing to repair, and the entire role is played by these MSCs. Again, they go back, anchor to the capillaries, stabilize the vascular system, endothelial system of the organ, and organ starts functioning back again normal. This is the beauty of this mesenchymal stem cell, which acts as a Chang's hypothesis, which says they play a major role, not only healing, but establishing a microenvironment of the healed organ to function normal also. This is a beautiful theory. What happens in the front side of MSC, when I'm showing my palm, in the front side of the MSC, usually when it is de-anchored from the capillary, the front side of the MSC plays a major role to secrete all immunomodulatory molecules. And protect the tissue from any kind of mycobacterium or any kind of bacterium or any kind of viruses or any kind of particles which are foreign, not to enter in the body, and try to segregate them and remove of the body using immunomodulators. MSC plays in the front side, and the rear side of the model cell gives a tropic effect of a repairing system, anti-apoptic, anti-scarring, angiogenic, mitotic. This is what basic function of a MSC plays here as a repairing cell. The MSC are the pericide and pericide are the MSC to some extent to understand this. But this is not 100% true. Let me tell you here the beauty of is that all MSCs are pericide, but all pericides are not MSCs. Because pericides, when they represent in different organs, like if you take a pericide of a capillary of a liver, pericide of a capillary of a kidney, pericide of neurons, brain, apart from the basic MSC definition of 3 specific markers, they also represent tissue-specific markers. That is a pericide, whereas MSC doesn't show tissue-specific markers. Whereas pericides show tissue-specific markers, that is CD markers, which are present in the liver. They have to show a specific markers which are specific to that liver. Whereas MSC may not show that. But all MSCs are pericides. All pericides are not MSC. Vice ratio is not correct. Very few scientists can understand this difference of pericide and MSCs, and which is coming up very fast in a newer aspect of regenerative medicine. This is a small video. If you can play, I'll try it. Okay, this video is not playing, but it will show you how an MSC, when it gets a tissue injury, releases different kind of cytokines, chemokines from front and back side, which I had described you earlier one, and that will show you a beauty of how the repairing is going on there. And this, probably I was trying to introduce this to make a concept of MSC to understand how 24/7 these drug stores or medical signaling cells are working in your body. So MSCs are medical signaling cells and not stem cells. They complete a complete regenerative microenvironment is created by MSC and very specific to that tissue healing. Very specific that tissue healing. If it's the skin, it plays a different role. If it's the liver, it plays a different role. If it's the brain, it plays a different role. Means every organ, it has got different roles to play. Now, if you see this MSC in a micro milieu, what does it do? Regenerate, maintenance of the tissue turnover required, bioactive factors, immunomodulatory, tropic effect. The beauty of it, the one cell playing almost all very much different roles at a time. That is the reason I call it to be a king of all the cells. Now, these cell produce bioactive factors, which I have already shown you, like anti-fibrotic, anti-scarring, angiogenic, mitotic. Now, as I was talking to you, there is one more role of MSCs, which earlier we were not able to find out about the exosomes in last 2-3 years. Actually, in fact, to tell you, exosomes were identified before the MSCs were identified. 1990s, MSCs came into knowledge, but existence of these exosomes, we, as such, the textbooks are showing, research book papers show they existed in the knowledge from 1983 onwards. They were before that known, but we were not knowing they are the chemical membrane molecules coming from MSCs. That is what is in the last 2 years we could find out. These are the membrane vesicles by way of exocytosis or way of endocytosis. These are small spherical molecules coming from the membranes of MSCs. They have got bipeptide, bi-lipid layer cover on the molecule surface. The range of size is from 10 to 1 micrometer, very small compared to the cell size. They come into different forms like apoptic bodies, which is responsible for deciding the fate of the cell, that is apopsis. They are the large size of exosomes, microvesicles, and macrovesicles. They all play a specialized. The beauty of these exosomes is that they are again specialized. Specialized means you can make exosomes specific to immunomodulation. You can make exosomes specific for apart from anti-cancer effect. You can make them for regenerative process. You can make for anti-inflammatory effect, the way other molecules are there. So 800 plus different. And let me share you, though I'm calling you today around, saying you around 800 plus, tomorrow, if you ask me to give a talk on MSc after a year, I would say rather, there could be another 200 molecules added, which we don't know today, which will come as we go deeper and deeper and deeper and deeper. We'll come to know what exosomes do play a major role as a membrane vesicles, and intercellular communication is maintained by exosomes. Intercellular communication. Apart from that, they also are responsible to remove the biological waste products from the cell by using a system called as heat shock proteins, chaperones. They also play a major role that proteins which are unfolded to remove from the cell. That also plays a major role by these exosomes. Now these exosomes, if I see you, is extracellular vesicles and intracellular vesicles. They play a major role in the form of producing various biochemical signaling systems from various functions of regeneration, immunomodulation, anti-cancer. Now it is a very clear picture about these membrane vesicles as exosomes. Now if you see mediated immunity, this particular slide will express to you about the exosomes from MSC, from a bone marrow, which can affecting the immune modulation of dendritic cells, because MSCs we call pan-immune suppressing cells. Pan-immune suppressing cells. Subsequently, I'll show you how it works to suppress the entire immune related from DC, NK, T to all B cells. They play a major role. And this comes through exosomes. This comes through exosomes. Now, the slide which I'm showing you, immunomodulation, if you see, here the molecules which are playing as a role in immunomodulations for dendritic cell, for B cells, for NK cells, T regulated cells. The way they are affecting on immunomass suppression. This is what you can read out from this slide. Now, if you see, apart from all the functions, I have written in gross here LL37 as a molecule. Now, how many of you know the MSCs produce antibiotic, antimicrobial? LL37 is one of the molecules of multiple antibiotic molecules produced by MSCs, which can target bacterium, can develop a resistance to the lab produced or factory produced antibiotic, but bacterium can never produce resistance to the antibiotic molecule produced by MSCs. They play a major role, recent role in management of COVID. MSCs play a major role, not only COVID, for any type of pneumonia, even tuberculosis, resistant tuberculosis, MSCs play a major role. If time permits, I can talk to you on that area also. This is the pan immunosuppression effect, which is shown. I may not take a lot or longer time. If anybody has student as a career this, they can all. One drug molecule has got action by MSC is multifold. There are many actions here. If you see, many actions like immunomodulation, anti-inflammation, anti-apoptic, cell differentiation, and regenerative potential. Now this slide, which is recently, actually, in fact, if you see, wonderful, before I started my talk today, right 15 minutes before, I included this slide for you to just show you a plethora of secretome of MSCs from various cytokines, growth factors, exosomes, macrocycles, having effect on cell differentiation, cancer exosomes, and tunneling microtubules in the way to develop microcapillaries. This is a beauty of MSC. How many could understand this drug molecule is going to be future of every medical specialty? When I talk of specialty, means specialty of cardiology, specialty of urology, specialty of neurology, specialty of dermatology, specialty of talk of a tissue, Bedside is there. Talk of a tissue, MSC is there. Talk of a tissue, the resident stem cell is different. Now this MSC makes the differentiation. Here I have shown cell differentiation. This differentiation is not of MSC. MSC don't differentiate into any specific tissue cells within the body in vivo. They will have an exosome, secretome coming to differentiate those tissue resident stem cells which are present in the niche of that tissue specific niches where the stem cells are present, and they play a major role to differentiate those specific different tissue specific cells and not the MSC themselves. This is what, please try to understand again. MSCs don't differentiate into any type of specific cells within the body, but outside the body, yes. Within the body, they are not stem cells. They are drugstores. They are signaling cells. They are medical signaling cells. So overall, if you see, one drug, one action in allopathic science, that is the reason I showed you the slide of Albert Einstein. If you don't change the concept of basic, you will never come to a solution to newer therapies which are biology based and not pharmacology based. Now understood? The pharmacological base is different and biological base is different. So we need to think with the new plate of knowledge which can give understanding of role of MSCs or biological molecules when we talk them as a drug molecule. So it is not a stem cell, it is a medical signaling cell. In between, people call is a stromal cell, but they are not also stromal cell. Neither they are stem cells. Stromal cells don't exist in the body. They are medicinal signaling cells. See, I'm trying to clear your basic concept about MSCs. So what is the additional feature for this beautiful cell? The additional beautiful feature of this stem cell is that MSC is that we can customize them as per our need. We can customize them as per our need. The need-based customization is very, very important. We can make in our lab very specific MSCs which are immunomodulating. TLR primed cells will be pro-inflammatory, like MSC1. I have shown you MSC2 here. If you remember, I don't know how many of them you know about the macrophages. One macrophage 1, macrophage 2. That's pro-inflammatory and anti-inflammatory. Similarly here, immunomodulatory and pro-inflammatory MSCs. You can customize them, personalize them, depending on the therapies, treatments, what you are looking for, using them into therapeutic molecule as you want. Now I'll come to a real question mark. When you want to say this particular cell molecule MSC, I want to bring this as a drug molecule, which I call as a living drug molecule compared to a non-living drug molecule. Making cell available as a drug is not so easy. Does it mean, is it so easy as the molecule of a pharmacological drug molecule? No. Why? The basic idea when I talk of a pharmacological molecule, it is a non-living. It is not affected by the environment. If I ask you to set up an antibiotic plant in Africa and ask you to set up a plant in US, both are developmentally at two different stages of life or developmental stages. But if you can understand, the molecule which comes as a final product in both the factories or both the places will be same giving effect, will not be different in biological efficacy because it's a non-living molecule. When I want to ask you, can you produce MSC in Africa with the same efficacy as US? Not so easy. Why? Because these are living, breathing entities. Stem cells or MSCs are the living, breathing entities. For them, we should have, when we are thinking of to make this cell into a drug therapies, it should be reproducible. It should be quality-wise reliable. It should be accessible to all. It should be cost-effective, because you can't have an expensive drug molecule to be used for somebody as a drug therapy, because it will go to the limited population. Again, when we are talking making available for millions and millions of people, the fundamental changes, it will be a very defined process, which, because I told you it's a living, breathing entity, it will change with every manipulation which you do in the laboratory. Person to person manipulation changes, the cell changes throughout the entire manufacturing process. Not only that, temperature, humidity, environment, timing, duration of you keeping cell into incubator, temperature within the incubator, gas exchange in the incubator, even oxygen concentration, everything affects the cell. So understood the difference between the manufacturing a cell as a drug molecule is much, much difficult compared to a drug molecule when we talk to be manufactured as a pharmacological molecule. So the entire concept is changing as a biology then now. Now here what we have to see is in the entire manufacturing process of a drug molecule like MSC, from harvesting, isolation, expansion, differentiation, transformation, packaging, cryopreservation, and distribution logistics system. Everything will be different, cannot be compared to a pharmacological drug molecule. So when we say cell as a product, it will be an empty promise if a manufacturing is not supported by sufficient, robust cell engineering innovations. Cell cannot be attributed as a drug molecule. So we need to establish very robust systems. So challenges what we are looking are reproducibility, reliability, scalability, stability of the molecule, quality of the molecule. Quality comes, what are the source from we are taking. If you take a MSC from a diabetic patient, the quality will be different of MSC. If you take a normal patient, MSC quality will be different. If you take a patient, aging patient, MSC quality will be different. If you take a young person donor, MSC quality will be different. And the most important, the cost. Are we in a position to have these kind of robust technologies by which we can make the people accessible to these type of products to reach to them? Yes, hurdles and challenges can be worked out very well. It requires a robust philosophy to grow billions of cells, which are more automated, robotic, controlled by sensors, to see that every stage in the incubator is monitored. At each stage, whether the cell of that batch can be preserved or not, is getting differentiated or not, at what stage these sensors will give information about them taking out of the incubators, going into quality assessment, and then going into an storage, cryo storage facilities. So all this can come through engineering advancement, manufacturing process, critical quality attributes we require. As I told you, various sensors as potency assays are required. To make it low cost, we have to make it robust technologies using robotic, flexible automations. And what is more important, when we talk of a cell molecule as a drug, supply chain management is very important, which depends on the raw materials coming in into the management of the factories and going out, which is very, very important. Now, clinical infrastructural advancements are very important. I believe, being students, I will not talk a lot on this. Let us go the goal of the future, deliver cell therapy to patients of a mass population, making them quality potent cells, reproducibility. And most important here, I would like to insist, since a new molecule it is there, we have to focus on a very important aspect of regulatory policies. And when we want to use as a drug, reimbursement policies from the insurance companies. These are very, very important aspects. Otherwise, they cannot become as a drug molecule. When I'm talking of a regulatory policy, if you see, regulatory framework in South Korea is different, Japan is different, US is different, India is different. So these are big challenges. We need to focus how intercontinental, inter-country, some common regulatory policies come up in future, so as they could be used as a drug molecule when manufactured in India could be supplied globally, manufactured in Africa supplied globally. At the same time, do we have any kind of policies? Can these treatments sit into reimbursement framework of the insurances? No, we still don't have. So we need a lot of these challenges to overcome in the application when we talk of a goal future when we want to take it. We at STEMRx, particularly having all approvals, we work on the institutional pattern. We have a lot of research work going on, particularly the MSCs. We have a GMP grade requirement. It jumped out one slide more. What is a public-private partnership? When we talk of stem cells, we require public partnership, means we require regulatory people, we require people coming from the engineering sector, we require people coming from the software sector, we are coming from the health sector, we are required from the academic sector. All these make these nascent industry absolutely de-risking and enabling them to take to a common man. At STEMRx, we follow almost every regulatory principle. We have all the approvals. We work a lot on MSCs, particularly my research on avascular necrosis is WRB approved, where we have around more than 700 patients done on avascular necrosis, various affiliations we have got, and various patents we have got. I think I will not extend beyond time. We already crossed the time. Anybody has got some questions on particularly pertaining to MSCs, you are free to ask me right now questions. I have tried to make a shortcut in some of the slides because the talk was extending beyond more than an hour, 1 hour, 15 minutes. The host can declare that my talk, that way everybody has listened very quietly. I'm thankful to all of them, particularly organizers, Dr. Indumathi, you made me to reach to all the students to understand the concept of regenerative medicine. See, only teaching is not important for the students. We have to understand them. How do we take to them to benches? So regulatory frameworks, their clinical applications, what are the challenges, how this MSc is going to change the entire future for medicine, entire future. That is what I always call MSc is the next leg of medicine to be practiced tomorrow by physicians, what medicines are practicing today. Thank you for a patient hearing.
Thank you, sir.
Thank you so much, sir, for such a wonderful, informative talk on mesenchymal stem cell as a living drug molecule. Hope this talk was definitely flourish everyone's knowledge about stem cell. Now ready for question session. Question answer session would be last.
Dr. Vijay Kumar Kutalas. He's the Secretary General. He's the Secretary General for FABA. So heartily I welcome Dr. Vijay Kumar Kutala also for this session, and thank you for being here till the end of this session, sir. We will now start with the discussion. I would just like to ask Dr. Vijay Kumar Kutala, sir, to say a few words.
Dr. Vijay Kumar Kutala, thank you for you joining the session. Everybody matters. So I'm really happy you waited till the last. Probably you have to remove.
You're in mute.
Microphone. The host can do that. Host can do that. Ask the host.
You're in mute.
Very good evening. Thank you, sir. You have a wonderful presentation on MSCs. Actually, MSc is also a very dream project for me.
Sir, you're again muted.
Sir, you're muted. Sir, please unmute yourself.
Yeah. Thanks for giving a new concept of MSCs. Totally new to me about new version of MSCs, what you're talking about. Really, it is exciting presentation. I really loved it. Thank you, sir, for giving. Definitely, we'll try to collaborate with your company. And I have one question, sir. You're talking about MSCs are new terminology like medicinally signaling molecule, say signaling cells, like any other cells. Like, you know, when you put it, when you inject the cells, due to autocrine effect, it get differentiated into other cells like that. But you're talking about that it is not going to differentiate. How are you going to support that, sir?
That is a very good question. I will make the concept of stem cell clear, basic of here. First of all, let me clear the concept.
Yes, sir. Yes, sir.
Has got a resident philosophy of every organ in a niche.
Yes, sir.
MSC is a parasite within the environment of the body. It is not a stem cell. When a stem cell is not when cell cannot differentiate, given an environment, it cannot be stem cell. And this is supported today by demonstrating in various animal models the markers using immunofluorescence technologies. MSC is getting out of anchored capillaries, doing the repairing jobs as the Chang's hypothesis, which I showed you, and the same MSC going back and anchoring to stabilize the new vascular system of the repair system, which is already demonstrated.
Yeah, if your claim is not stem cells, so why so much restriction from regulatory bodies to restrict the use of MSCs all over the globe?
Yes, again, now very nice question when it comes in the frame of regulatory framework.
Yes.
Any biological molecule coming from the human source and using for therapeutic comes under by law as a drug.
Yes.
And we don't have any kind of done studies till date apart from few of the MSCs used in specific indications from GVHD or myocardial in the country like Japan and South Korea, nowhere else. So the fate of this cell also, I'll add for few, the beautiful future of this also. Yes. How many of us know what is the fate of MSC when given the body?
Exactly, yes.
What is the fate of this cell?
Yes.
This is again a big question mark.
Yes. Yeah.
So till we solve the fate of the MSC is that they are rejected in the body after doing their basic job of repair, wear and tear within 7 to 10 days. So the body rejects them. Being a microscopic molecule, the rejections are not realized by us, but they are rejected. So regulatory framework has to come up for these molecules. Reason being that if not regulated, there is a misuse of these molecules and we really don't know where they are effective, how much they are effective. So everything which comes as a drug molecule has to go through a process of animal human model.
Yeah, actually, a lot of clinical trials, especially in Europe, where people are using for hematopoietic stem cell transplantation and also renal transplantation. MSCs are doing extremely good in prevention of GVDH and also renal rejection. So can you?
I will open a new box for you now here as far as I have studied. Very good new box. Very few scientists may be knowing it.
Yes.
Plethora of secretome by MSC in immunomodulation.
Yes.
This is the only cell in the body. I repeat my sentence. Only cell in the body which plays a role in every organ, every tissue, every system from immunomodulation to regeneration. There is no other cell in the body which has got a universal role. And that is the reason, if you see, I have shown one of the slides, MSCs play a major role in pan-immunosuppression.
Yeah, I've seen it, yeah.
That is the role where you want to reduce the immunosuppressive drugs and steroids, you need to use MSC. And that too, again, I'll make it still more clear. When to use MSC, when you want to use it as an immunomodulation in organ transplant.
Correct. Yeah.
It has to be given at a time when organ is being transplanted. At that moment, when the antigenicity is being exchanged.
Yeah.
If you give before that, it is useless.
Correct.
If you give after that, after 2 hours, by that time antigenicity is already established.
Yes.
Got it now? You have to give at that transmission phase where the body is taking up a new organ.
Yes.
That is a moment if you give, you will have beautiful post-transplant benefit.
Correct. I accept that. Yeah.
Transplant, I'll add further more knowledge to you. So since your interest is there.
Yeah, sure.
Chimeric transplants using entire immune system change of a donor, recipient. Chimeric bone marrow transplant along with the organ transplant. This is last 10 years being studied. It has come to clinical side now and will come in future. The biology will change the future. I think it's the fifth new leg of the medicine for the reason this particular technology will rule out use of any immunosuppressants and steroids in future for organ transplant.
Yes, sir. Thank you. Thank you very much, sir. Thank you very much.
Sir, Professor Vijay Kumar Kutala, thank you so much for your active participation and also sharing your thoughts in this session. Nice to have you, sir.
Yeah, please go ahead. Thank you, Lord. Thank you.
Now we will start the discussion. Yes, sir. We'll start the discussion session. Questions asked by students. Leena, please carry on.
Yeah.
Leena?
Yes, ma'am.
Please carry on.
Thank you so much, sir. Hope this talk was definitely flourish everyone knowledge about stem cell. Now ready for question session. May I now request Dr. Som Subranath to moderate this session. Please.
Dr. Som Subranath? Dr. Somsubranath, are you in? Dr. Nath, are you in? Dr. Nath, Leena, you can just start with the first question that was asked by. Leena, you can start. You can start asking the first question asked by students to sir. Sir, actually, we because of the creativity of interest to the students, what we have done is we are every week selecting 2 best students, and the criteria is one based on what relevant questions that they ask to the speakers, and second criteria from your webinar we would be selecting 10 questions and giving them a Google form. So like last week we selected 2 best students. So students are already posted their questions on chat. So I would request Leena to ask the questions one by one, and it would be nice if you could answer that. Please. Leena, carry on.
First question is from Fatima. Can MSC be used for treatment of neurodegenerative disease by reducing the degeneration of neurons and aggregation of protein?
Very nice question. Whoever the student is there, I should appreciate her thinking ability, particularly in degenerative conditions. When we talk of degeneration, particularly neurodegenerative conditions, the new concept of management of neurodegenerative condition is called as unfolded protein response. Yes, the question has a validity. They can be used in these conditions, neurodegenerative, particularly you can talk of Parkinson, Alzheimer, or supranuclear palsy progressive PSPs. These are called as the neurodegenerative conditions where these MSCs work through their plethora of exosomes. The system within the cell where unfolded protein responses which are working through their heat shock proteins, those are called as chaperones in common language. These chaperones do make a lot of difference with the MSCs being used. Yes, it can be used in almost all types of neurodegenerative diseases. I hope you got the answer.
Now proceed with the second question.
But the first question's answered. Is she okay?
Fatima?
Yeah, she's replied. Yes. Leena, please proceed. Thank you, sir. Please proceed with the second question.
Second question from Netra Deshpande. How do we identify exosome release by MSC and other different cell types?
This question is a very nice question. Again, the students are well oriented. Probably they have heard the talk very nicely. Now, the exosomes are the membrane particles one has to, and they are containing various type of RNAs, not only the proteins, mRNAs, miRNAs, DNAs, which are to be marked before we separate them. And using the technologies of supersonic, sonic ultrasounds, or using a filtration technologies, this can be isolated. And presence of the specific indication of mRNAs or RNAs or DNAs or protein molecules, we can identify them as exosomes. And we can, with specific indication also, we can identify them as exosomes. And every cell has got a different range of exosomes. Like adult cells do also produce, but the range is again different, which is to be marked with the, again, RNA studies or protein studies.
Yeah, Leena, proceed with the third question.
Next question from Nishan. Can MSC will beneficial to repair lungs tissue which damaged by COVID-19?
With the current scenario, if you see, there are a lot of studies currently in the clinical practice. Now, when I will add this question to the question with the earlier one, Fatima, what was the girl's name? Both the questions I'll add together here, where I'll put together answer which will be giving two concepts. One, per se MSC are of size of 10 plus microns, larger than the capillaries. The entrapment of the MSCs after IV infusion is in the lungs, first entrapment for 24 to 40 hours. in the microenvironment where is inflammatory, COVID response is beautiful, and definitely they can be used 3 purposes. One, immunomodulation, anti-inflammatory, and regeneration. Normally post-COVID, if you see, the people are coming with fibrosis of the lung, fibrosis, ILDs, interstitial fibrosis, because of the decreased compliance and elasticity of the lung. So where MSCs play a major role, yes, they do use. The second part of the exosome of the MSC. Now, if you see the recent study which was done in Israel, which is still under much consideration, and I'm very much proponent of this therapy. Even if you use MSCs intramuscular, I repeat my sentence, even if you use MSCs as intramuscular, they release all type of required exosomes and take these exosomes to the site where the inflammation is to be controlled and repairing job is to be done in the lungs. Yes, they have done it and they have proved it, and it is working very well for COVID intramuscular as well as intravenous. In intramuscular, it is an exosome, and in intravenous, it is an entrapment of MSC within the lung where the paracrine effect and cytokine effect can be realized.
Yeah, Leena, go ahead.
Thank you, sir. Next question from Janvi. What happens if pericytes are not anchored to capillaries?
Probably she was sleeping while I was talking. Pericytes are always anchored to capillaries. That is a basic place, basic function they have got in the body to be anchored on the capillaries, nowhere else.
Leena, please ask this question about Niharika's research on MSC in India and the antibiotic releasing, Parul Sharma. So there was a question by one of the faculty. What about the research of MSC in India? And there was another question by one student called Parul Sharma. MSC as a next generation can MSC be the next generation antibiotic releasing entity?
Very nice. Both the questions are very good. Here, I'll tell you, when we talk of antimicrobial action, yes, MSCs will replace a lot of antibiotics in the future. Even myself, I have realized and
So your voice is breaking. So your voice is breaking.
Can you hear me now?
No, we are getting some background music. Can somebody mute your this thing?
Let them mute their
Can
microphones.
somebody mute your microphones, please? Sorry, sir. Please continue.
Fine. Now, antibiotic secreted by secretome of MSC changes with the antigen of the bacterium. Remember one thing. It doesn't remain the same secretome, because these exosomes which I spoke just now are the membrane particles, mRNAs, RNAs, which are changing depending on the antigenicity and the environment of the microbacterium or any bacterium. So we will never have resistance to the antibiotics produced by antimicrobial proteins produced by the MSCs. First answer to this question, antibiotic. And second was, what is the second question?
The research of MSC in India.
Yeah. In research of MSC in India, if you try to go to a clinical trial GOV, we have a very limited research on MSCs in India compared to what is happening in Western and US continent and Korea and Japan. If you see the research involved, because research requires 2 important concepts. One is a government support, second is money, and basic researchers. These 3 are very limited resources in India. The MSC's research in India has got a limited scope, but still we are coming up now. I would say rather there are very few clinical trials in India there on MSCs compared to what we have got in South Korea and US, particularly Japan, where I have one workup going on with MSC in Japan.
So in addition to this question, we also have in which country MSc are more done? MSc therapy are more done. In which country MSc therapy is more done?
US.
Yeah, the next
Active
question.
work is in US. Today,
Lina, could you please take the...
today
Sorry.
I would say rather, the university where I'm visiting faculty as Indianapolis University and hosted university, in these both universities, there's a huge amount of budget spent on MSc research. Huge means huge. We just can't imagine. And various animal models are there for particularly GVHD, wound healing, diabetes, for using exosomes of MSCs, type 1 diabetes, much being used. Then even there's an technology which is called, I will recollect the name, which is done by a Chinese scientist whom I know very well. He is also from US. T-educator therapy using the stem cells from the cord blood, MSCs from the cord blood. T-educator therapy, T-educator therapy, which are much ahead of we can even think of, particularly exomes.
Thank you, sir. Lina, please pick up the temporal and spatial release question.
Yes, ma'am. Next question from Niharika. Can MSc use in individual treatment therapy, especially cancer treatment? And next question, similar question. How can MSc be used to treat severe skin burns?
In both the questions, the ideal is the environment of the tissue. When we talk for malignancy, MSc in the exogenous source are not advisable to be used in the malignancies. One. Second, there are studies where MSCs are used as an drug delivery systems, because MSCs cross blood-brain barriers. So for the neuroblastomas of the brain, MSCs to deliver specific targeted drugs are molecules, chemotherapeutic molecules have been used in malignancies. But as such, MSCs are not advised to use in the therapeutics. But exosomes can be used separately as for malignant treatment, anti-malignancies, as a drug molecule. In when we use MSc in the lab, but not as MSc in the body in vivo. And in the wound healing in burns, they make wonderful, as I told you, they reduce the fibrosis, scarring, as well as the new tissue regeneration, and healing time is reduced.
Thank you, sir. Next question from Vishal.
Temporal and spatial question. You have the question, no, about temporal spatial. Because due to lack of time, we'll just choose the selected questions. There was a question about temporal.
Yes. How question from Parul Sharma. How does the temporal and spatial release of microvesicle induce the microenvironment required for differentiation of other stem cell in it?
What she wants to ask this question, I am really not connecting. Again, you can read out this question. Temporal.
How does the temporal and spatial release of microvesicle induce the microenvironment required for differentiation of the other stem cell in the knee?
Temporal area, fascial area, what is that? What
release temporal and spatial release of microvesicles induced.
You can probably, she can redo it again, skip the question and go to the next one.
Whether it's an anatomical area, fascial and temporal you are talking about?
She asked, how does the temporal and spatial release of microvesicle...
Could you please reframe and resend your question? Meantime, we will address other questions. Please reframe and resend your question. Go to the other question.
Markings of the face and head.
Other question.
Please go to the other question.
Other question come from the Asha. What can be done to make MSc produce in different countries and under different conditions same? How can we prevent them from being different?
Probably the entire concept is based on the living, breathing entity. we cannot, the question is wrongly framed. Let me tell you, whether the MSC is being produced in US or in African country, I compared there with showing that the difference between a living drug molecule, breathing molecule, it keeps on changing from a person who handles in the laboratory with the micropipette, in the environment with the gas exchange concentration, temperature, humidity, to the storage and the duration of the time. We have to have their all standardizations done first. And that can be done through sensors, robotic technologies, and software technology. That is the reason when I said when you want to manufacture these kind of molecules as a drug, it is an integrated program from different specialties coming together, from a software, from electronic, from a robotic, you got to understand now, and then from the biotechnology and from the medicine, then the technology can be established as a small, what you can say, village where these systems can be established to establish a standardization of manufacturing of the stem cells as a drug molecule. Otherwise, they are different, absolutely different. In the same lab, same person doing in different places, same person. I repeat my sentence, same person given at 2 different times. If not knowing importance of every step and not standardize, yield of stem cell MSC at 2-3 different sessions will be different. Their secretomes will be different. Their concentrations and shapes will be different. And the results will be different otherwise, if they are not standardized. In the same lab, they will be different.
Thank you, sir. Next question from myself. As per my knowledge, even in stem cell therapy, immunosuppressive drug is given in case of allogenic sources. Here I wanted to know, is immunosuppressive drug is needed in MSC-based therapy? Because as we know, MSC is known to be an immunomodulant.
MSC, Lina, very nice. I'll ask you, we'll make you a little clear here now. Very nice. See, MSCs are, first remember, immunomodulatory. They are not as you said, they are not immunomodulatory. It's not. They are pan-immunomodulatory drug or cell molecule, first. Second principle. Yes, immunosuppressants are used in stem cell transplant, but they are used in hematopoietic stem cell transplant. Because HLA-DR and MSC class 1 and class 2 are representing hematopoietic stem cells, not the MSCs. MSCs, they don't represent, first of all, major histopathology complex 1, they don't represent. Very weak MSC, major histopathology complex 2, they represent, but they are HLA-DR negative. So they are very, very mildly immunogenic, very, very mildly. That is the reason they survive in the body for a longer period, 7 to 10 days, I spoke during my talk. But they do.
Thank you, sir. Next question from Nimisha Gaikwad. What media can be...
directly go on to Ms. Sujahan's question. Sujahan. We'll just pick up, I'll just tell you, Ms. Sujahan had asked 2 questions. You can pick any one or you can pick both. Please scroll down, you'll find Ms. Sujahan's question.
Sujan.
Yeah.
Yeah. When you say that MSC produce specific antibody respective to the antigen presented, I would like to know if APCs can present antigen to MSC.
My dear student, antigen presenting cell comes from a category of immunogenicity. Their job is to represent the foreign antigens on their surface. They don't have any role in antibiotic management.
No, the question was if you don't present the antigen, how would the MSC recognize which anti produce?
Again, I repeat my sentence. MSCs keep on changing their molecule. I told you they produce 800 plus different kind of protein molecules. Tomorrow, after a year, if you talk to me, it would be 1,000, I said you. The reason being that they have never been used in antimicrobial in a clinical aspect. They are being tried clinically now, and they have shown at least more than 40-50 types of antimicrobial activities seen by them with different kind of microbiomes. The most resistant tuberculous bacteria, which are existing today, resistant microbial patients, which are resistant to standard medical care of drugs, are also sensitive to the microbial action produced by MSCs. So my here question is not the antigen, and bacterial antigen is not produced by APC cells. Let me clear first of all, those the viral, they are, bacteria itself is sufficient for to have microbial action. APC doesn't induce any antimicrobial action in the MSCs. First, let me clear you. Antigen presenting cell don't induce any antimicrobial action in the MSCs. It is a local microbial environment which bacteria carries which type of protein, which is the antigen. Accordingly, the MSCs will produce the antimicrobial protein. Not because the APC is representing. I think I made clear now.
Yeah, I got it. I got it.
We will stop with last two questions. One question a student has asked, Mr. Sadaf, can the MSC be used as a vaccine for different types of diseases?
No, they can never be used.
And another question by Ms. Drisha. As you said, MSC can be customized as per requirements. So why not the research solution start from the approach? Does this have any drawback?
In fact, the drawbacks will only come into existence when we go to the research. Right now, in the current state, MSCs, when they are being customized, the customization is for them to produce specific type of secretones. When we want antimicrobial, when we want anti-inflammatory, when we want immunomodulatory, when we want regenerative, those type of particular specific proteins we can induce in the MSCs to produce immunomodulatory effect or regenerative effect. But currently, these are all in trials. Clinically, they are not used. The animal models are there, definitely.
So there's another last question. I think sir spared really a lot of time in discussion. I would just like to request you to answer the last question. The person who asked the question, she's reframed the question, sir. She's just reframed the question in such a way: can release time and gradient of signaling molecules affect the function that signaling? Just a second. Can release time and gradient of signaling molecules affect the function that signaling molecule perform?
No, repeat the question.
Can release time. If Carol, can you just switch on your microphone and ask the question yourself, because even I'm not able to get the exact question. Can you have reframed the question, but can you, if you don't mind, switch on the microphone and ask yourself the question?
Yes, ma'am. Sure. Ma'am, am I audible?
Yeah, you're audible. Go ahead.
Good evening, sir. So my question is, like as you said that there are various signaling molecules released by our MSC cells. So so the time of release and the concentration in which they release, does this affect the function they are going to perform? Means the kind of response they are going to affect on the stem cells residing in the niche?
Parul, the question, two questions before your question which was asked about the MSCs being produced at different places, why do they change their outcomes? Now here the answer to you is the same way. Yes, they do affect, depending on the release, where the inflammatory environment is there, where the immunomodulatory environment is there. The concentration and release time do affect the outcome of any kind of disease. Very correct. It affects.
Okay, sir. Sir, just last thing to say, sir, I'm actually a final year student who is in BTech Biotechnology, and I'm definitely passionate enough to pursue my master's, and I'm thinking to pursue my master's in this field, regenerative medicine. And so before listening to your lecture, I was like all over the internet. I was searching through going through research papers, and most of them nowadays were focusing on iPSC cells. Whereas after listening to the lecture today, by the presentation, it feels that MSCs are more promising. And the thing which you told about their antimicrobial activity and the unexplored aspect of it, would you advise me to whether should I take up this topic? Because I'm finding it very interesting, because earlier also I've worked on AMR. So that is the reason.
Parul, I'll explain you. Remember one thing, take in bold writing in your diary. iPSCs ever, never will come to clinical site, first of all.
I agree with you, sir.
Never, never come. They will always remain in the laboratories only. First principle. Second principle, MSCs will come the next generation drug molecule to change the practicing medicine by the physicians in future. These two things, please note down in your diary. To add further ahead, remember one thing, the science is evolving very, very, very fast. What I spoke just now is a bit of what I will tell you now, a very innovative concept. Now, future is coming with the concept of body, your own body acting as a bioreactor. Not the laboratories, not the incubators. We can manipulate the cell in your own body to change the function through using various technologies of, like you can say, what you call viral vectors. You can use particularly this electrical waves, sonic waves, to induce these changes using various mRNAs, miRNAs, and various protein molecules. Now, right now, we will be conducting the world's first human trial, where the somatic skin cells will be induced to differentiate into neurons and endothelial cells, where body will act as an incubator, as a bioreactor. Because the environment what body can give, nobody else can give. Those things with new mRNA technologies which are coming up, exomes are coming up, will change the future. How do I repair my organ within the body itself? This is the future for all of us.
Yeah. Sir, lastly, there was a multiple question related to MSCs and cancer. I've not looked specifically because there were many people who were asking about mesenchymal stem cell therapy for cancer and what is the effect of mesenchymal stem cells to treat tumorigenicity, tumor progression. So how would it actually be a cure for treating cancer? And with this, we would really end the session. You have really had enormous patience. So please do answer this final question, and definitely we would relieve you after this.
Only thing, it will be worthful if I have given added something best to you, all of your students, which probably if you search out thousands of papers, you will not get what I have given you today. What is more important here in malignancies, when we talk of MSCs, there's a clear understanding. MSCs we should not use in any kind of application in the malignancies. First of all, because there is a very dubious role for stem cells to be used in the malignancies. Second, there is no role as such. Only thing, there are few studies which I told you earlier on. As a delivery targets, MSCs can be used for the drug delivery. in cancers, in malignancies. Some studies have been done and they have proved to be really good, with good outcome, particularly in neural malignancies where blood-brain barrier has a restriction to pass the chemotherapeutic molecules. Otherwise, MSCs I'll not advise anywhere except immunosuppression in bone marrow transplants or organ transplants. For immunosuppression, that too. I hope that makes sense.
Yes, sir. Leena, please conclude the session.
Thank you so much, sir, for very informative and interactive session. Hope everyone cleared their doubt here. We apologize if any question is remaining. We got lots of questions, but due to shortage of time, we have to end this discussion here. Now I request Miss Nayan to give vote of thanks.
Yes, Leena. I hope I'm audible.
Yeah, you are.
Thank you, ma'am. Thank you, Leena, for giving me a moment to extend my vote of thanks. First of all, a big thank you to our honorable guest and speaker of today's session, Dr. Pradeep Mahajan. Thank you, sir, for sharing your precious time with us and for such an informative and interesting session on MSCs. Also, I'm very sure all our participants must have had a wonderful learning experience. So once again, thank you very much, sir. I also extend my thanks to all our participants for your efforts to make this session interactive. I also thank Dr. Pankaj sir for being a wonderful coordinator of all our sessions. I also wish to express my gratitude to Dr. Som Subranath sir for being an excellent moderator of our sessions. Although I think probably due to some technical glitch, we couldn't connect with him today, but thank you, sir. I also thank Ms. Leena for her amazing coordination and also for her moderating the question answer session. So thanks, Leena. And last but not the least, I would like to thank Dr. Indumathi ma'am, Director of IABM, for organizing such wonderful webinar sessions every weekend. So thank you, ma'am, and thank you everyone for making this session successful. Thank you.
So finally, there was one question, career in stem cell. I hope students are so connected to you that they're not in a position to leave you. Could you please, don't mind to add a point on career in stem cells for the sake of students? Please, sir.
In fact, for all of you who are right now are connected to the subject here, let me share with you, when I said you, the next limb of medicine or next leg of medicine are these stem cells. So my dear students, regenerative medicine, stem cells make a small part of entire regenerative medicine. They are going to be future for all of you, whether you want or don't want, but they will change your future. I would rather welcome all of you with a focus, what you want to do, which area you want to select. There cannot be a universal application. You have to select the application, you have to select the subject and focus on that. There is much to do compared to any other subject which is already done. Here, everything is unexplored. You have a chance to swim as much as you want. Thank you, Indumathi.
Thank you, sir. I hope this is the only session where even after vote of thanks, we make the speaker, speak. But that's really kind of you and really nice of you. Thank you so much for that. And I would really glad and I hope that we would collaborate for upcoming events, and that will be a wonderful platform for all our IABM students for, upcoming any programs, internships, trainings for collaboration with STEMRx, and looking forward to it, sir. Great having you on this platform. Thank you, Nayan, for the vote of thanks. Without taking much time, declare that this particular webinar is over. And thank you overall, one and all, for your participation, because again, the thank list will go on, and let's make sure that sir is relieved right now. Great having you.
Thank you.
Thank you all. Bye-bye. Good night. Have a great weekend.